On February 23rd, 2020, I gave two lectures at the SUNY School of Optometry as part of a symposium that was organized by Murray Fingeret and Richard Madonna. Um, and this is the first lecture, and the second will be also available on my website as a separate video. So before I start, let me just go over the obligatory financial disclosures. In addition to NEI grants, I have support in the form of grants as well as equipment from Topcon and Heidelberg, and that support is relevant to these lectures. So as I said, there'll be two lectures. The first lecture, I'm going to deal with the question, what can OCT teach us about the nature of glaucomatous damage? Then the second lecture, it'll be more practical. I'm going to take up the question, what are the implications for the clinician? All right, so let's just start with some background anatomy. I'm going to remind you that ganglion cells are killed by glaucoma. So glaucoma kills retinal ganglion cells and their axons. So let's just review some anatomical terms so that we're using the same terms. So this indicates a piece of the retina at the back of the eye. The retina is about 300 microns thick. Here's an electron micrograph of a human retina. And 3 layers of cells. You've got the cell bodies for the receptors, cell bodies for the bipolars and horizontals and amacrine cells, and the cell bodies for the ganglion cells. So light comes in this way, of course. It's absorbed in the outer segment here of the receptors, and then they make synaptic contact with the bipolars. That makes synaptic contact with the ganglion cells. And it's the axons of the ganglion cells that leave the eye as part of the optic nerve, and you have about 1 million ganglion cells and 1 million optic nerve fibers, or axons. So receptors, internuclear layer, then the layers that are important to us. The inner plexiform layer, where these synapses take place, retinal ganglion cell layer, and retinal nerve fiber layer. And the retinal nerve fiber layer contains the axons of the ganglion cells. It also has blood vessels and glial tissue. All right, so again, glaucoma kills retinal ganglion cells and their axons. So this layer is affected, as is this, as is this. So these are the layers that are damaged by glaucoma. Excuse me. Okay, now, to understand glaucomatous damage, you also have to understand the route that the retinal nerve fiber layers, that is the axons of the ganglion cells, take as they travel to the optic disc. You've all seen schematics, such as this one. It's meant to capture a few key factors of the surface anatomy. First, ganglion cells on the nasal side, that is on the disc side of the fovea, their axons go more or less directly into the disc. The ones on the temporal side, the opposite side of the fovea, their axons arc around and avoid the fovea, and they bunch up here where they meet with axons from other parts of the eye, and this is a particularly vulnerable place for glaucomatous damage. Same thing up here. I'm not drawing in all the different bundles, but same thing, that's a vulnerable area. So this is a nice illustration of that. This is a person who was born with some of their axons myelinated. I'll remind you that the axons of the ganglion cells do not become myelinated in the normal eye until after the lamina cribrosa. So this eye had myelinization before that, and you can actually see, because of that, the arcuate nature of these paths that the retinal nerve fiber layer bundles take. Okay, now, local damage, because damage occurs at and in the disc, at the lamina cribrosa, you're going to get these characteristic arcuate defects from local damage. Here's another example. All right, now why is that important? Well, another thing I want to remind you of is that there's no single litmus test for glaucoma. That is, glaucoma is traditionally defined as a progressive optic neuropathy, that is a disease of the optic nerve, that's characterized by a particular pattern of anatomical, historically anatomical meant fundus exam, or maybe with a fundus photo. Increasingly, it's becoming associated with OCT as well as a fundus exam, whereas behavioral changes refer, at least for the last 20, 30 years, to visual fields from automated perimetry. Okay, so by specific pattern, we're going to mean arcuate patterns. So I want you to keep in mind that we're going to be looking for patterns that are characteristic of a glaucomatous damage. We're looking for patterns. We're not looking for global statistics or thickness, right? Ultimately, we want to decide whether we have patterns that are characteristic of glaucoma. All right, so next question. What can you see on the OCT scans? What can they actually see? Well, here's a scan through the back of the eye, and that's the OCT scan. Let me remind you that you can think of this as OCT as being like ultrasound, but with light. And because of light has a shorter wavelength, the resolution is much better than ultrasound. All right, so you're seeing about that much of the back of the eye here. So just to draw this, if this were a ganglion cell here, that would be its axon. So that's the retinal ganglion cell plus inner plexiform layer. That's the retinal nerve fiber layer made up of the axons. Now, typically, you'll be looking at a circle scan or a derived circle scan. I'll say more about that in a moment. So here's the results of a circle scan for the same eye, right? So what you're doing here, right, that's retinal nerve fiber layer. And you're looking in, you're looking in this way, like this. Does that help? Whoops, excuse me. That is, you're looking in this way here. All right, now, over 10 years ago, there was just one machine. And the machine was the Zeiss Stratus time domain machine. And we all looked at one report. This is the report we looked at. Right? That scan, by the way, started in the temporal region and went around clockwise. This is what the image looked like if you blow it up. You wouldn't know when you looked at this image how well this machine did. Right? This is often called a T-snit curve because the scan over here went from temporal to superior to nasal to inferior to temporal. That is T-snit. Right? Here is an image now with the newer. And when I say newer, the images like this have been the images have been this good for 10 years or so, 8 to 10 years, ever since these spectral domain machines. Okay? They're much better than the old time domain OCT images. This is much better than that. This, of course, is in pseudo color. All right, again, that's retinal nerve fiber layer. This plot that we often look at, the dark curve here is called circumpapillary retinal nerve fiber thickness plot, and that is, or profile sometimes. And what this is this is the thickness here of the retinal nerve fiber layer. Okay, so let's take this example. Here's an eye. You can actually see the defects on this eye, on the fundus photo. Let's look at a circle scan from this eye. Here it is. This is a circle scan. This is retinal nerve fiber layer again. This is what a healthy would look like. So you can actually see the defects here. Let's take this much here, the center part. This is, by the way, the center. This is going to be this part of the disc here, because this is now an end-stent. I'm going to be showing you from now on largely end-stent plots. I'm going to be showing you the circle scan or derived circle scan as if it's the scanning started here and went counterclockwise. The reason for it is it puts the temporal half here that you're most interested in, temporal half here, right smack in the middle, rather than dividing it in half. So let's take this and flip it. So now you can see, this is this, might be, and that would be there. So here's what an axon might look like. So you can see that this healthy part corresponds to this here. We can actually flip it, it might be easier to see. This healthy part here corresponds to that. That's what the bundle here would look like, for example. All right, so you can see remarkable detail. To get a better understanding of what you could see, a number of years ago, we decided to compare the OCT images we get to adaptive optics, what you would get with adaptive optics. So I don't know how much of you know about adaptive optics, but it's actually a technique that comes out of astronomy, and it's by taking images, it's a light technique. By taking images with deformable mirrors, you can correct for the aberrations of the eye and get these extraordinarily detailed pictures. So here is a study that was done in Richard Rosen's lab with a piece of equipment built by Alf Dubra, and Toko Chu here was the person who did all the hard work here. So this is the adaptive optics image. This is actually made up of a number of 1 degree by 1 degree images. Here, this, by the way, took Toko about, I don't know, 35 minutes to obtain, and took people in my lab 2 days to analyze. 2 days. This is not a clinical test. This is the spectral domain OCT. This took 2 seconds to obtain and a few seconds to analyze. And what you're looking at, this OCT is more or less along this curve here. So we can line this up now. Here are the blood vessels, and here are the shadows associated with those blood vessels on the OCT. So we know where we are. So I want you to notice, excuse me, wherever there are remaining bundles, there's remaining tissue. Bundles, tissue. Bundles, hyperreflective region indicating axons, and so on. And wherever this is dark, it's hyporeflective, right? And so on. So these hyperreflective regions are preserved retinal nerve fiber bundles. So you can see regions of preserved bundles on the OCT. It's remarkable. The message here is most clinicians, by not looking at these images, the result is that we are not making optimal clinical use of the information in the OCT. All right, finally, what are the lessons one can learn from the OCT? First, let's talk about where the damage is occurring around the disc. So the data here come from a study that we published about 7 years ago now, and we had data from both control eyes, healthy control eyes, and also patients that were either suspicious or had glaucoma. Everyone had cube scans, 2 cube scans, one centered on the fovea, one centered on the disc. Each of these cube scans is made up of 128 individual line scans. So there are 128 line scans here. This would be one of them right through the fovea. And on this, you can see the retinal nerve fiber layer, and you can see retinal ganglion cell plus inner plexiform layer. And whenever I say retinal ganglion cell layer, in this lecture, I'm actually going to mean retinal ganglion cell plus inner plexiform layer. And same here. From this, we're going to get retinal nerve fiber layer. All right, here's a pseudo color plot where dark red is very thick and blue is very thin. Okay, so what regions of the disc are most affected by glaucomatous damage? That is, where do we expect the circumpapillary retinal nerve fiber layer damage due to glaucoma? So let's take a look at retinal nerve fiber layer thickness color map again. This is controls, and now I'm looking at a smaller number because I wanted an age-matched group for my patients. That's thin, that's thick, and let's blow it up and center it on the disc. Remember, from classical teaching, these are the regions that are particularly vulnerable. So if this is superior disc, the superior quadrant is vulnerable, and the inferior quadrant is vulnerable. This was nicely shown in the early 80s by Quigley and colleagues, and there's more recent data as well. All right, so here's the average now for 23 eyes with moderate glaucoma. So, but moderate glaucoma meaning that their mean deviation on the 24-2 is somewhere between -6 and -10 or something. So, if we take the difference between this, we get this. Now, I apologize for this, although it's not my fault. Red means different things here. Green means not thinner than normal. Here, red means much thinner. You can picture very significant normal, where here it meant thicker. Let's blow it up and I'll go over it again. On this difference plot, this is within normal limits, and dark red is extensive thinning, and the colors in between are in between. All right, so extensive thinning. All right, so if we superimpose it on the traditional view, we can see that it's largely in agreement, except that instead of the entire quadrant being particularly vulnerable to glaucoma, it's the temporal half here and here and here, these temporal regions that are particularly vulnerable. So what's new? The temporal half of the superior and inferior quadrants are the most vulnerable to glaucoma's damage, not the entire quadrant. So here and here. Okay. So the question we were just dealing with is what regions of the disc are most affected, right? Well, what about the macula or central retina? Right? So as part of the study, we also had cube scans centered on the fovea. And from that, we can get retinal ganglion cell thickness. And dark red is very thick, dark blue is very thin. And all of this thickness here is within about 8 degrees of fixation. So I'm going to define macula. If you look at the literature, different specialties define the macula in different ways. For me, it's going to be plus or minus 8 degrees, and it's the region with the greatest density of ganglion cells. In fact, this region here represents only about 2% of the retina, but has over 30% of your ganglion cells. And it's the region that's so important to read, drive, recognize faces, and so on. All right, so let's take this and now replace this with ganglion cells in the macula. So this is retinal nerve fiber layer thickness for the controls. Here it is for the patients. Here it is the difference between the two. And again, I want to remind you that dark red means extensive thinning, green means within normal limits. So let's superimpose this here. This is retinal ganglion cell loss. This is retinal nerve fiber layer loss. That's your macula. All right, what's new? Now we have something new. What we found was that early damage includes the macula. All right, so here's a figure from a paper in 2011 in which we were looking at damage to the macula, and here's here's our anatomical model that summarizes the results. So this region here fits the traditional thinking that the temporal quadrant is not particularly vulnerable to glaucoma. All of the ganglion cells here go into the temporal quadrant, and as we were taught, the temporal quadrant is less vulnerable. However, so that's less vulnerable. However, contrary to traditional thinking, these this region is very vulnerable to early damage, and it's because the ganglion cells here send their axons right smack into the most vulnerable region of the disc. And we call this the macular vulnerability zone. So here it is over here, right? That's the temporal, and this is the macular vulnerability zone. So why does early damage include the macula? Well, there are two reasons. First of all, local defects often include this region here, but there's also widespread, early, very subtle damage that takes place as well that we now know about. And here's one study from Dana Blumberg and colleagues that describes that. All right. So early glaucomatous damage includes the macula and is very common. That's one of the take-home messages. So how common? Well, here's a study we did a couple of years ago in which we had 57 eyes with early glaucoma. And here I'm defining early glaucoma in the traditional way based on visual fields. So it's a 24-2 mean deviation better than -6 dB. Okay. So in this study, we had glaucoma specialists that we gave them all the information we had, and they judged whether these eyes had glaucoma or not. 57 did. Of these 57, damage to the macular vulnerability zone occurred in over 3/4 of these eyes with early glaucoma, whereas damage in general was even higher, close to 88%. So 3/4 had damage in this region, 88% had damage that included that region plus the rest of the macula. Essentially, and all the eyes had damage either up here and or down here. All right, so to put this in perspective, I want to compare our view of what's happening to the traditional model of early glaucomatous damage. So traditionally, you've got the superior region that's vulnerable and the inferior region that's vulnerable. And the temporal region is less vulnerable. So the assumptions of I'm going to claim that the assumption of the traditional model is first that superior and inferior quadrants are the most vulnerable. Well, we largely agree with that. The answer is yes, but it's largely the temporal half. Second assumption of the traditional model is that the temporal quadrant is less vulnerable. And again, we agree. Yes to a first approximation. This is the most vulnerable region, followed by this, followed by this. But early damage can include diffuse temporal damage. Third, the traditional view is that the axons from the ganglion cells in the macular region enter the temporal quadrant of the disc. And this is where we differ. Again, no, right? What we've found is that the axons from this part of the macula enter into the inferior vulnerability zone. And then fourth, the implicit model came to concludes that macular function is minimally affected in early glaucoma. The answer is no, not true. In early glaucoma, you can very often see arcuate defects due to damage in the macular vulnerability zone, and then there's also this diffuse damage I mentioned. Okay, third, there are implications here for categorizing early glaucomatous damage based on OCT. So I want to do the following thought experiment with you. Assume that OCT came before visual fields and automated perimetry. Maybe you knew about Goldman's, but you didn't have automated perimetry. But you had OCT. How would we categorize glaucomatous damage? Right? Well, I'm going to argue it'd be something like the following. If we had high-resolution circle scans here that have been available to us for 8-10 years, right? Let's look at this part here that's associated with the central part of your field, right? So we're going to look at this part right here, right? Like that. So I'm going to argue that first of all, you would you would notice that the location of the defect. So there it is. There's the defect there. Right here it is on the thickness plot. And there it is there too. So you've got two defects here on the thickness plot. You probably categorize the severity based on depth, let's say. So if we blow that up, you can see this is pretty deep. There's not much retinal nerve fiber layer remaining here. Here's another eye with damage exactly in the same place. Very, very common place. Damage exactly in the same place, but now in this particular eye, it's less severe because it's not as deep. Right? There, compared to that. More affected, less affected. Okay, next width. So here's a third eye that again is very deep here, but look how wide this is, right? It goes from about here to about here, right? So location, depth, width, and then local variation. So in this particular eye here, for example, it's essentially gone here, the retinal nerve fiber layer. It's markedly decreased, but there's little left. It's decreased here, but there's a lot left. So, and again, that would correspond to these locations. That white line arrow corresponds to this. This plot here corresponds to there. And that location corresponds to there. Again, you can actually see these local regions of preserved. So this is essentially gone. You should have no vision associated with that probably, but you'll have a little something here, more there. All right, so what are the implications? Well, one implication is you should expect a large variety, infinite variety, in fact, of circumpapillary retinal nerve fiber layer defects. All right. So let's just take a look at this region here within this box. That region will have damage that varies in location, depth, width, and then local variation. And that location corresponds to about that much of the disc. This is what we've called the inferior vulnerability zone. That's the macular vulnerability zone. All right, so here's the image that's associated with this eye. Here are four other eyes that I didn't work very hard to get variation. The variation is just there, but you can see these eyes all have damage in that same region, but it varies in terms of location, depth, width, and then local variation. Okay, so finally, how can I compare abnormalities on the OCT to defects on the visual field? Well, here's a classic categorization of visual fields. And we'll come back to that in a moment. First, let me just go back and remind you that there's no litmus test, and we're looking at specific pattern of anatomical, right, and visual behavioral changes. And by behavioral changes, we mean visual field. Okay, I think you all are familiar with this report, but let me just remind you that the subject here is pressing a button every time they see a light, and the lights are very brief and very small. All right. But most importantly, I want to remind you of how crudely the sampling, spatially crude, the sampling is in this report. So here's your macula. Here's the region that's so important for vision. There are only 4 points here because the test points here are spaced every every 6 degrees, starting up and over by 3 degrees. So you only have 4 of these within 8 degrees. These numbers, again, I want to remind you, the way you read this is a -13 here, for example, means this person at this spot is 1.3 log units less sensitive than normal. Or if you take the anti-log, it means they're 1/20th as normal. Or if you think of it the other way around, they need 20 times more light than you do. Right? If this was a -3, it would be 0.3 log units, and yet they would need twice as much light. And then, of course, these symbols here signify the significance level. So one thing we'll use is the mean deviation here, which it's not quite this, but you can think about it as the average of these points. Okay, this is the most common visual field test used to diagnose glaucoma. So visual fields are often used to categorize patterns, right? So here is a scheme developed for the Oates trials by Keltner Johnson and colleagues, right? And they illustrate their categorization based here on upper visual field defects, or at least I'm illustrating it that way. So this is a nasal step. This was called paracentral defect. This a partial arcuate. This an arcuate. This an altitudinal defect. So how do we relate these local changes in damage at the disc to visual field locations? Well, you need a map. And here's a map that relates the location of the 24-2 points to the location on the retina for an average eye. All right, so here are your test locations. Right? By the way, this is a 30-2, so there's an extra ring of points here. All right, so this will help us relate one to the other. So let's just consider these defects for starters, right? What I want to point out here, is these defects here all fall within this region here. Right? That is, they fall within here and that part of the disc. So we can take this little model and superimpose it, keeping the eye correct. This was in the left eye. That's the right eye. That's the left eye. In any case, this is the model superimposed, and you can see how where these points are falling in relation to this region here. Even this eye here that has more extensive damage, that's the model. That means this damage out here is now over here, around here. So let me pause for a second to let you absorb this, and let me put it another way. All of these defects here, all the different, in all their variations, they are largely due to damage within this region right here of the disc. That's it. Right. Okay, and there's your macular vulnerability zone. Okay, so again, all of those defects correspond to, you get all the different variety of defects here due to damage in this one region here of the disc, right there. Okay, so what are the implications? We should expect not simple classifications, but a wide variety of patterns of visual field loss. So these defects here should be associated with a wide variety of visual field defects because of all this variation you can have. And so, in fact, here are the visual fields for these particular eyes with the model. Again, and damage is going to include the macula. All right, so that's the main part of what I want to describe here. In the second lecture, we're going to turn to what are the implications for the clinician. But let me just say in passing, so I wouldn't have to I may repeat some of this in the second lecture, but the problem here is that the traditional way of looking at in the clinic, where you do a 24-2 and an OCT disc scan, you can miss or underestimate macular damage. So here's the location of your 24-2. Here is what you're getting in with early to moderate damage, right? And you can see that these 24-2 points poorly sample this region of damage. And here are just some of the studies that have been done supporting this. So the common clinical tests that are done, which I'm going to argue are typically a 24-2 visual field and OCT disc scan, right? You we're going to argue in the next lecture that you're going to want to include visual fields with finer grid, a 10-2 or modified. And you're going to want more than just the cube scan of the disc. You'll want a cube scan of the macula as well. Thank you, and I hope you will, if you need to review this lecture, play it again, be my guest, and I hope you'll tune in for the second video. Thank you. Oh, before I end, let me just remind you that you can actually go to the website, which I suspect you're already on if you're listening to this lecture, and not only hear this lecture, but other lectures, as well as obtain these review articles that we have. Thank you.